Let's talk about muscles and muscle contractions. Before we can understand the muscle contraction, we need to look at the structure of the muscle. Muscles are made of bundles of muscle cells. So here's a muscle, and inside there are bundles, and those bundles are bundles of muscle cells. If we look at that bundle and look at a single cell or a muscle fiber, and we zoom in on that muscle fiber, we see that inside this single muscle cell or muscle fiber are many myofibrils, or muscle filaments, units of contraction. If we pull out one of these myofibrils, or muscle fibers, and zoom in on it, we see that it has this very distinct banding pattern with these dark lines called Z-bands. These Z-bands mark the beginning and end of each individual contraction unit called a sarcomere. The sarcomere is a contractional unit, and it's composed of an array of protein fibers called actin and myosin. The actin is a thicker fiber, and the actin is a thinner fiber, and the myosin is a thicker fiber, and they're arranged in this very specific array. And as these fibers slide past one another, this unit of contraction, the sarcomere, can condense, get shorter from Z-band to Z-band line. So, if we think about how a muscle contracts, we actually have to think down to the actual protein filaments of a muscle fiber, the contraction unit of the sarcomere, this array of actin and myosin, and as the actin fibers slide past the myosin fibers, the sarcomere will contract. Well, if this Z-band to Z-band distance contracts, as does this one, then this whole muscle fiber or myofibril will contract. If many myofibrils contract, this muscle cell will contract. And if many muscle cells contract, then the bundles of muscle cells or the muscle will contract. This ability of this tissue to contract is distinctive of muscle tissue alone. To look at how this process works, let's go a little bit deeper. Let's zoom in on a single muscle cell or muscle fiber. You have the plasma membrane here on the outside, and this muscle cell is packed full of myofibrils, or these filaments of contraction units. There's a sarcomere, Z-band to Z-band, Z-band, Z-band. We have our array of myosin and actin fibers. Also, you can see inside this muscle cell, let me grab a highlighter here, are many, maybe a different color will work, lots of these organelles. What do you think these organelles are? Jammed up here in the sides, we have many mitochondria, which makes sense because we're burning lots of fuel in this muscle cell. But if we look at this myofibril, wrapped around this myofibril, we see this network of tubules, kind of containers of membrane, and they're housing lots of calcium ions. So we have our muscle cell jam-packed full of protein filament and arrays. Here's a Z-band to Z-band, so there's one sarcomere. And wrapping around those myofibrils are these sarcoplasmic reticulum, or these containers that are holding calcium ions, or Ca++ calcium ions. And the muscle cell is going to contract on a signal from a motor neuron. So we need to go back to remember a little bit of the nervous system, and I have drawn here the end of one neuron, and here's our synapse, and this forms what we call our neuromuscular junction, the junction between a motor neuron and a muscle cell. And so we might receive an action potential moving down the axon, and as that action potential moves down, calcium ions come in, and as those calcium ions come in, we have the vesicles move to the synapse and dump their neurotransmitters in. And since this is a neuromuscular junction, these neurotransmitters here are called acetylcholine. Acetylcholine are the neurotransmitters of the neuromuscular junction. And these neurotransmitters will cross the synapse and bind to receptors on the surface of the muscle cell. Now, to see what happens next, we need to go a little deeper. As these neurotransmitters bind to receptor sites on the surface of the cell membrane, they're going to depolarize the cell. That depolarization travels inside of the cell and causes the sarcoplasmic reticulum to depolarize, and when it does, it releases its contents. And if we remember, the sarcoplasmic reticulum contains calcium ions. To see why the release of the calcium ions is important, we need to look even deeper. Zooming in to the individual filaments that we see, way down deep in here, we zoom in, and here's an actin fiber, the thin fiber, and here's a myosin fiber, a thick fiber. And the myosin would like to bind to the actin, but it can't because there's a molecule called troponin or tropomyosin that's blocking the binding sites. And the way to move this tropomyosin out of the binding sites so that these little gripping hands from the myosin can connect to the actin, we need calcium. When calcium is released, and calcium is represented by these black diamonds here, as calcium is released, it causes the tropomyosin to move out of the binding sites, these white binding sites. And once we move that tropomyosin out of the way, these cross bridges from the thick myosin fibers can bind to the actin fibers, and then powered by ATP, these little heads, these myosin heads that are crossing over, will change shape in what's called a power stroke and pull against the actin. Another ATP will cause it to release, and then it will reach up and pull again, and slowly pull its way down this actin fiber, bringing it to the middle. We have a short animation that's going to help make this more clear. Okay, so here we go. Here's our muscle cell. There's our sarcoplasmic reticulum inside. Here are our array of actin and myosin. We zoom in. Here comes the active potential, releasing the acetylcholine, which depolarizes the muscle cell. That depolarization transfers through and causes the release of calcium ions in the sarcoplasmic reticulum. Then we zoom in closer. Right now, the actin can't bind, the myosin heads can't bind to the actin because tropomyosin is away. But if we add calcium, the tropomyosin moves out of the way, powered by ATP. The myosin heads can form cross bridges, and then with another ATP releases and has the power stroke. And we slowly pull against that actin, and we're drawing our sarcomere, our Z-bands, closer together. If we decrease the calcium, the tropomyosin move back, and the muscle relaxes. So, if we were to put this in a step-like manner, and put like step 1, step 2, step 3, it might look something like this. Step 1, impulse from motor neuron reaches the muscle. Step 2, the sarcoplasmic reticulum that stores calcium releases calcium, which diffuses into the myofibrils and reaches the actin filaments. Step 3, calcium ions move the tropomyosin from the blocking sites, binding sites on the actin. Step 4, cross bridges from the myosin can attach for the power stroke, powered by ATP. Step 5, another ATP input causes the cross bridges to release and then reattach for another power stroke. This is a very energy-expensive process. ATP is used rapidly, and the mitochondria that are packed into those muscle cells are breaking down glucose to provide energy. However, we need a source of phosphate to attach to the ADP. Creatine phosphate is a naturally occurring substance in the muscle cells that donates the phosphate for this process. For the first 5 to 10 minutes of activity, the muscle cell uses its stores of glycogen as a glucose source for aerobic respiration. For the next half hour or so, the muscle depends on glucose and fatty acids delivered by the blood to be broken down for respiration, and after that, fatty acids. During this time, oxygen debt in muscles could cause the muscle cells to switch over to glycolysis alone. The ATP yield is low but fast, and the lactic acid can build up. The more efficient we are in delivering oxygen to the muscles, the longer we can stay in an aerobic respiration state. So, for the 1-minute review, muscles are made of bundles of muscle cells. Muscle cells contain an array of cables called myofibrils. Myofibrils are a series of contraction units called sarcomeres. Z-bands distinguish where one sarcomere stops and the next one begins. Each sarcomere is an array of filaments, the thin actin protein filament and the thick myosin. These filaments slide past each other in what's called the sliding filament model of contraction. In the process of the muscle contraction, the neuromuscular junction, acetylcholine, is released. This excitatory neurotransmitter depolarizes the membrane of the muscle cell, which causes the release of calcium ions from the sarcoplasmic reticulum. The release of calcium ions from the sarcoplasmic reticulum move a protein called tropomyosin off the binding sites on the actin filaments, allowing the myosin cross bridges to form. Powered by ATP, the myosin heads pull against the actin, drawing the sarcomere together.